Bismillahirrahmanirrahim. Today I am going to discuss excitation-contraction coupling mechanism in skeletal muscle, and I will also discuss transverse tubules and sarcoplasmic reticulum system. So what is excitation-contraction coupling mechanism? The term excitation-contraction coupling, which is written in short as ECC, describe the rapid communication between electrical events occur in the plasma membrane of skeletal muscle membrane and calcium release from the sarcoplasmic reticulum, which leads to contraction. It means that two different events by two different structures sharing their effect at one common point and produce different action. Structures are neuron and muscle fiber. Events of action potential and depolarization of muscle cell or muscle fiber membrane, and common point is neuromuscular junction, and output is contraction of the muscle fiber. So coupling of these events is called excitation-contraction coupling. Excitation-contraction coupling consists of two linked phases. One is the excitation phase and the other one is the contraction phase. Excitation refers to the generation of an action potential within the muscle cell. The electrical signals that lead to muscle cell contraction begin as a nerve impulse in the neuron and is relayed to the muscle via motor neuron. Here you can see this is the motor neuron in which action potential occur, and it is relay or arrive at the muscle fiber membrane. The connection between a motor neuron and a muscle cell is called the neuromuscular junction. You know that this is a motor neuron fiber branching fiber, and this is the muscle fiber, and the junction between these two, the muscle fiber and the neuronal fiber, is called neuromuscular junction. So it is the common point at which these two events actually share, the muscle and the nerve share these events. Here impulses are transmitted between the nervous system and the muscle, propagating the action potential that will lead to muscle contraction. So this is the point from which action potential is transmitted to the muscle fiber, and this causes contraction of the muscle fiber. We have discussed all these in the neuromuscular junction. Full details are available in that lecture if somebody is interested, so he can listen to that lecture. Now the contraction phase. Contraction refers to a physical change to the shape of the muscle cell, also called a muscle fiber. So in relaxed form, the muscle cells are actually long, and when they contract, they become shorter. So when a muscle fiber contracts, it becomes shorter. Here you can see this is a muscle. When it is relaxed, the belly is long, because this belly is composed of these muscles fiber, and when these muscles fiber are relaxed, they are long. But when they contract, these fibers contract, the whole belly contract, and the muscle fiber become shorter in size. Purpose of excitation-contraction coupling. Why we need excitation-contraction coupling mechanism? Because excitation-contraction coupling is vital for the movement of all muscle in the body. For example, heartbeat, breathing, blinking, walking, and even our ability to move food through the digestive system. Ingestion of food, its mixing, its propulsion through the digestive system, and digestion. For all these mechanisms, we need excitation-contraction coupling mechanism. Now, transverse tubule and reticulo-sarcoplasmic reticulum play a most important role in the excitation-contraction coupling mechanism. So, we will discuss these structure in detail. T-tubule lies adjacent to the end of longitudinal sarcoplasmic reticulum tubule that surround all sides of the actual myofibril that contract. We have two structures. One is the T-tubule. T means transfer and T-tubule means transversely located. They are transversely located, and the other structure is sarcoplasmic reticulum, which are located longitudinally, and they are actually the myofibril. This is the structure which actually contract. We have discussed that it contains actin and myosin, and when these actin and myosin overlap each other, the muscle become contracted. So these two structures actually cover this myofibril. The T-tubules are located transversely, and the sarcoplasmic reticulum. This is the sarcoplasmic reticulum. It is located longitudinally. The T-tubules are very small and run transversely to the myofibril. They begin at the cell membrane and penetrate all the way from one side to the muscle fiber to the other side. This is the muscle membrane or sarcolemma. So these T-tubules originate from this muscle membrane and muscle fiber membrane and transversely and on the other side of the these muscle fibers on the opposite side. Communicate with the extracellular fluid. Where the T-tubule originate from the cell membrane, they are open to the exterior of the muscle fiber. This is the muscle fiber. It is covered by the sarcolemma, and from sarcolemma the T-tubules originate. These T-tubules have opening in the sarcolemma, and by this opening the T-tubules are actually communicate with the extracellular fluid. Therefore, they communicate with the extracellular fluid surrounding the muscle fiber, and they themselves contain extracellular fluid in their lumen. They are communicated with the extracellular fluid, and these tubules are filled with the extracellular fluid. On the other hand, they cover the muscle fibers. T-tubules are actually internal extension of the cell membrane. They are actually part of the cell membrane. They originate from the cell. They have the tissue of the cell membrane. And the electrical current spread on muscle fiber membrane and also in T-tubule and then elicit the muscle contraction. So this tubule is the invagination of the cell membrane around the muscle fiber, and when the depolarization of the sarcolemma or muscle membrane occur, the T-tubule are also depolarization also occur in these T-tubules, and this causes muscle contraction. Sarcoplasmic reticulum. This is composed of two major parts. Sarcoplasmic reticulum. This reticulum is composed of two major parts. Large chamber called terminal cisternae that about the T-tubules. Here you can see that they have cisternae or chambers in the area where they terminate near the T-tubules or in the proximity of the T-tubules. They have chambers of cisternae, and these cisternae are filled with the fluid, and long longitudinal tubules that surround all surface of the actual contractile myofibril. These are the longitudinal tubules. Here this is the transverse or cisternae. Here you can see the sarcoplasmic reticulum tubule when they terminate near the transverse tubules. Here they make a cisternae or chamber filled with the fluid, extracellular fluid, and they have longitudinal tubules, and these tubules hold these myofibrils. Release of calcium ion by the sarcoplasmic reticulum. One of the special features of the sarcoplasmic reticulum is that within the vesicular tubule is an excess of calcium ion in high concentration, and many of these ions are released from each vesicle when an action potential occurs in the adjacent tubule. Here this is the sarcoplasmic reticulum. Remember, this is the sarcolemma, these are T-tubules, and this is interior of the cell where these are the sarcoplasmic reticulums, and these are the end of the sarcoplasmic reticulum, which are cisternae or chambers. In these chambers, actually high concentration of calcium is present. So when the calcium is released due to action potential when they are stimulated. How they are stimulated, we'll discuss it in the next slide. A muscle fiber is excited by a motor nerve. They generate an action potential. They spread along the surface membrane, sarcolemma, and transverse tubule system into a deeper part of the muscle fiber. Remember, the muscle fiber of skeletal muscle are thick, so the action potential cannot spread to the whole fiber or around the whole fiber, so the action potential is transmitted through these T-tubules. So when the action potential spread, it also spread around these T-tubules. In the T-tubules, a receptor, protein dihydropyridine, which is called DPH. Here in the T-tubule we have a protein receptor which is called dihydropyridine. Since the membrane depolarization, here the membrane is depolarized by the action potential, and this depolarization spread along the T-tubule, this receptor sense this depolarization and activate another receptor which is present on the cisternae of the sarcoplasmic reticulum, and these receptors are called ryanodine receptors. They open the calcium channels and release calcium from the sarcoplasmic reticulum. They bind to the troponin and activate the contraction process. So here we know that on the actin molecule we have troponin, and when this calcium is released, it attached to that troponin and it discover the G protein active site for the myosin head, and myosin head attached to it and cause contraction of these fibers. Calcium pump for removing calcium ion from the myofibrillar fluid after contraction. When contraction is over, then the calcium is removed from the myofibrillar. By this mechanism, muscle contraction continues as long as the calcium ion remain in the high concentration. When calcium is available for the actin and myosin in high concentration, the muscle remain in contracted position. However, a continually active calcium pump located in the wall of the sarcoplasmic reticulum pump calcium ion away from the myofibrillar back into the sarcoplasmic tubules. Here we have a pump which is called a calcium pump, and this calcium pump remove the calcium from the calcium ion away from the myofibrillar. They remove the calcium from myofibrillar to the sarcoplasmic tubule back. This is a energy dependent pump by which calcium is taken back into the sarcoplasmic reticulum. This pump can concentrate the calcium ion about 10,000-fold inside the tubules. In addition, inside the reticulum is a protein called calciquestrin that can bind up to 40 times more calcium. Here we have a receptor which can bind. This receptor is known as calciquestrin, to which calcium is attached, and 40 times more calcium can be concentrated in the sarcoplasmic reticulum. So these are the stages: the action potential, the extracellular fluid, intracellular fluid, and when the action potential arises, depolarization of the membrane occurs. This depolarization spread also over the T tubules. Here the L type or DHP type sensors activate, the calcium channel are open, calcium from the T tubule enter into the muscle fiber, and also by activation of the ryanodine receptors, they open the calcium channel on the endoplastic reticulum, and calcium also is come out from the endoplastic reticulum. Both these calcium cause a spark. They sum, and this summation cause a spark. This spark is summoned to the reef are attached with the troponin on the actin receptor, which is present on the actin molecule of the myofibril and activate the myosin head, to which myosin head is attached, and contraction of the myofibril occur and it become shorter. This is in the relaxed form and this is in the contracted form. Then after, when the contraction is over, the calcium is then retaken by the sarcoplasmic reticulum by ATP form, that is a calcium form which is energy dependent, and also calcium is taken out from the myofibril to the extracellular fluid by exchange with one sodium, and one sodium is taken in and 3 calcium are bring out from the myofibril, and the sodium potassium balance is maintained by the sodium potassium pump. So this was all about excitation contraction coupling mechanism. So thank you. Please subscribe the channel and press the bell icon for upcoming notification. Thank you once again.